in multiple different forms of limb-girdle muscular dystrophy. You may recall that early gene therapy programs back in the 1990s had bad immune responses because they were using adenoviruses. Here, adeno-associated viruses, these are out there around, they're in the environment, they're largely don't cause problems for humans. And so that's why that particular virus has been adopted for most of these gene therapies. So this is the kind of the nuts and bolts of what might go into one of those viral vectors. You have a promoter, so that's the part of the gene that turns the gene on. That's the light switch that turns the lights on and off in the room. When companies are designing gene therapies or investigators, you can make that light switch on all the time. You can make it so that light switch only turns on in the bedrooms. You can make it so that light switch turns on when you say, into your Alexa or Siri that turn on the light. There's just a wide variety of those promoters, and really that's an important step because that's what allows that gene to be expressed effectively into the muscle cells. So if you have you can imagine if you're missing a gene or a protein, you want something that allows that gene to be really well expressed in your muscles that are missing it. You have the transgene itself, which is the here in most cases is the gene that's missing, and then you have kind of the end polyadenoylation sequence. A really important point of gene therapy as it stands today in 2021 is that the maximum length is 4.7 kilobases. So for those of you who are old enough to remember burning CDs or DVDs from your computer or even on a flash drive, there's only so much that you can fit onto that. And so that's going to be an important thing. I'm going to come back to that maximum length when we talk about various different forms of limb-girdle and how people are thinking about it. We can go to the next slide. So then the other piece that you need to think about, or the investigators thought about, the clinical trialists and physicians thought about, and you should know, is this viral vector. So Dr. Matthews spoke about this as well, that many gene therapies, for limb-girdle muscular dystrophy, you want a AAV vector that can get into the muscle. And so this is a list of naturally occurring AAV vectors. And many development programs in limb-girdle muscular dystrophy have used either AAV9 or AAV8 or even AAV7. And you can see if you look at what target organs they go to, that the reason why you're choosing that as opposed to say AAV2 is because you want a virus that goes to the muscle. That's an important thing. You may want a virus that goes to the heart, like in LGMD-R9. So AAV9 might be more preferential than, say, AAV7, for example. But one real important caveat here is that you'll notice that every single one of those naturally occurring vectors that goes to the muscle also goes to the liver. Liver is not an affected organ in the limb-girdle muscular dystrophies. And the more virus that you accumulate in the liver, as we've learned from spinal muscular atrophy, the more toxicity, the more somebody can have side effects or problems from getting the virus. And so the real balance in designing these clinical trials is to make sure that you give enough of the virus to get the gene to the muscle, but not so much that you cause problems in the liver. And that's an area of intense research right now. There's a lot of active development in that. And well, I'm not talking about today, these are naturally occurring viruses. The synthetic viruses that people are designing to fix that problem in a lot of ways. They're designed to get more virus into the muscle at a lower virus level and less into the liver. So at the end of the day, that's going to be an important discussion point. When you hear people talk about clinical trials that are coming, you may hear people talk about this viral vector issue. Next slide. We talked a little bit about the promoter. I'm not going to spend a lot of time about it, but you should just know that, when you're getting a gene replacement therapy, people are thinking about a promoter that is turned on in the muscle, that it gets to highly expressed genes, and it's really the main driver of gene expression. So, the efficacy of the gene therapy depends on the ability of the virus to deliver the gene to the muscle and how well that promoter turns that gene on. So at the end of the day, those are really kind of two important levers we have to making for more effective gene therapy. Next slide. All right, so here we talk about the types of Carvers. So as it stands right now, it tends to be capped at 4.7 kilobases of exogenous DNA. And this is a big deal in limb-girdle. I think for smaller genes like the sarcoglycans, it's not a problem. You can fit the entire gene in there. Even for Fkrp, you can fit the entire gene in there. For genes like dysferlin, for example, that by itself is too big of a gene to fit alone in one virus. And so one thing you can do, which is shown on the figure here, is to give half the gene in one virus and give the other half, the back half of the gene, in another virus. And if you put the right kind of coding sequences on it and you give both viruses at the same time, when they're in the cells, they can come into they can combine to rejoin and create a sufficient length of the dysferlin protein or gene, for example. So that's that dual vector approach. Other approaches like in Duchenne muscular dystrophy is to trim the sequence down into the minimal important length. So it's like a microdystrophin, which has been tried, like I said, in Duchenne muscular dystrophy. For some of our limb-girdle muscular dystrophies, like those affected by titinopathies, for example, this would require multiple viruses at the same time. So I think some of those larger genes, this is going to be an issue we're going to have to encounter. We're going to have to figure out a way to get beyond that. And so if you're thinking about the genes that are first up in terms of gene replacement therapies, those are one of the reasons for why that might be is because they involve a gene mutation that is in a smaller gene, and so it's easier to deliver that. And so I think, when you're, like I said, when you're speaking to the investigators or the companies or the clinician in front of you, that's a real consideration depending on what form of limb-girdle muscular dystrophy you have. Next slide. We talked about a little bit some of the other kind of limitations of gene therapy. Liver tropism, so the fact that these gene therapies go to liver may lower the amount of gene replacement that you're able to receive. It may have issues with the overall weight of a person and the ability to give the right amount of gene replacement. There is, as we've learned from spinal muscular atrophy, as Dr. Matthews alluded to as well, there can often be a transient immune reaction. I think at this point, again, based on the learnings there, people have a pretty good handle on how to try to deal with that. And then I think there's an open question about durability. As it stands, like I said, in 2021, the idea is that you can really only give one of these gene therapies once because you do generate an immune reaction following that viral delivery. And the idea is that gene continues to exist in the muscle cells and it continues to produce the missing protein. But your muscle cells divide, they replicate, and so whether or not that's durable over a lifetime of a person is an open question and will require future study as we continue to move forward with these therapies. I think the other limitation that I didn't speak to is that largely what we've been talking about is that you're missing a gene and that you are able to deliver that gene that you're missing back. And that works pretty well for a lot of the recessive forms of limb-girdle, so LGMD2 or LGMDR. It works less well for some of the dominant forms of limb-girdle muscular dystrophy, so LGMD1 or LGMDD. And I'm not going to talk about it today, but I think in the future, knockdown approaches using this type of approach may be possible. Next slide. Okay, so let's look at some data. People have made lots of headway with these types of gene therapy. This is data looking at a particular gene therapy for beta-sarcoglycan. And there you can see perhaps the best figure is there on the right, where you can see that the untreated mouse muscles are on the left side of that panel and they're kind of dark and you don't see any protein expression. And when you deliver that gene therapy, you're able to see the return of that sarcoglycan beta expression. And the figure on the right shows that it goes into lots of places that you care about. So it goes into the muscles, it goes into the heart, it goes into the breathing muscles. And so that's, it's quite promising data. Next slide. And that in those same mice, that's great that they can show that they can return expression of the protein, but do they get better? Do they get stronger? And here are two measures, one looking at strength and the other is looking at muscle fatigue. And what you can see in the far bar or the treated mouse, that opposed to either the knockout, that they're substantially stronger and less fatigued than the knockout, and really actually quite close to what a wild type mouse would be. So it's great, as we have, and I think people have said before, it's great that we're able to effectively show that we can treat a mouse. But I think, whether or not this is going to be effective in the human is an open question, and something that I think most of us are pretty excited to start to understand. Next slide. This is probably one of, if not the most advanced gene replacement program across the limb-girdle muscular dystrophies. This data is taken from the Sarepta website, so you can go to the same website I did and look at it yourself. And what they showed in their early phase clinical trial, in that a very initial cohort, they were able to show that CK was reduced, so it was there's less sign of muscle damage. They were able to show that the beta-sarcoglycan expression was improved, which is what you want to see, and that functional measures were improved. So all very promising data from that early phase clinical trial as it stands. So next slide. So I think we're really at the precipice. This is kind of like, you're just about to turn on the fire hose here with all of these different programs, which is just super excited for, I speak for myself as a clinician and investigator. I know it's exciting for the patient community as well. Dr. Matthews alluded to or spoke about the Fkrp gene therapy programs that are coming in the near term, likely in the next year or so. But beyond that, you can see kind of the list of different genes that are targeted either in clinical programs. So these are people are doing early safety studies in beta-sarcoglycan, alpha-sarcoglycan, or dysferlin. Or if you look at these companies' websites, you can see that sarcoglycan gamma, ANO5, calpain-3, and Fkrp are all in the near term being targeted for gene replacement programs. So that's a lot of hope and optimism that we can get over the hump for these particular subtypes of limb-girdle muscular dystrophy. And I know that we will learn a lot through doing these clinical trials as a partnership with you, and that hopefully we become more efficient and continue to design better gene therapies as we go. Next slide. A brief word. So the nice thing, and Dr. Matthews alluded to this as well, it's kind of in the same bucket as gene therapy. So cell therapies are also in early development across the limb-girdle muscular dystrophy. So skeletal muscles, in theory, have substantial amount of regenerative capacity. So when you exercise and you break down muscles, you're breaking down muscles, you're growing new muscles. So how can we harness that regenerative capacity, that natural regenerative capacity of skeletal muscles? The problem, of course, in limb-girdle muscular dystrophies is that those muscles still have the same gene defect. So one idea is that you can take the kind of the satellite cells, or these are the cells from which you grow new muscles, and you can take them out and put in, correct the gene defect and put them back in, and that will allow those muscles to regrow. So this has been ongoing work in oncology. So there's a roadmap here to follow. It's a bit cumbersome as it stands right now, and I'm not going to go into details about issues around kind of your body's immune response to foreign cells. But there is early development in LGMD 2A or R1 for a similar type of therapy. Next slide. And this just shows that there's a model for this in Duchenne muscular dystrophy. So people across the muscular dystrophies are thinking about restoring satellite cell populations, and that would allow you to kind of regrow and regenerate a healthy skeletal muscle. So lots more to come. I think there's a lot of hope and optimism. We're reliant on your participation, on your partnership as part of these clinical trials as they proceed on the foundational natural history studies and patient registries that develop the tools to appropriately study and develop these new exciting therapies. And of course, just our fingers crossed that we can see some success here. And that's everything I have.
Thanks, everybody, for those excellent presentations. We're now going to move into the question and answer portion. My first question is actually for Dr. Kang. So if somebody was to participate in a trial, would that make them ineligible to participate in another clinical trial?
That's a really good question and an important one for patients to keep in mind. Natural history study trial participation or natural history study participation generally doesn't disqualify you from participating in a clinical trial. However, if you are receiving a potential new treatment in a clinical trial, there are times when that may interfere with participation in a future trial. And the rules vary depending on the exact two trials involved. Sometimes you just have to be off the first treatment for a while to be eligible for the second one. Sometimes if you've gotten the first treatment at all, you can't participate in certain other trials. And it doesn't mean that you can't get the therapy later on if it's FDA approved. There are different rules for that that'll be established. But the goal is to get the most clear data possible. And so that's the goal of the investigators, as well as to make sure that they're observing the safest conditions possible.
Great. And a quick follow-up question to that. If somebody was to join a clinical trial and change their mind, are they allowed to leave?
I believe that it's generally required that any consent form has a provision for a patient to leave the study. However, there are instances where, for example, with a gene therapy trial, if you've already received the gene therapy, it's typically a one-dose administration, as my colleagues have mentioned. Then, as Dr. Johnson has mentioned, you can't generally take that back. So you do need to be monitored. However, if you are in a trial where you're receiving a medication or a new therapy that requires, say, a weekly infusion through an IV or taking a pill once a day, that is something that you can stop if you really feel that you're experiencing unwanted side effects, or if there are other reasons that that have triggered this change in your decision. There are provisions for that, and even if you withdraw, the investigators will be monitoring you for safety to make sure that everything is okay. It does cause some problems in terms of data analysis and interpretation of the trial when patients withdraw, especially if a lot of them do. And so you do want to think carefully before you sign up and be ready to make that commitment. And that's why I mentioned that you should ask questions about how invasive it is, how much travel is involved, all of these things that will give you an idea of what the true commitment is. But yes, in answer to your question, there are pretty much always provisions for withdrawal, and that you'll be taken care of if that becomes necessary.
Thank you. My next question is for Dr. Johnson. I know that a lot of people are excited about investigational clinical trials because they want to be the first to get a potential treatment, but there's a lot of concern about placebo groups. Some people really don't want to be put into one. So can you speak a little bit about what placebo groups look like for LGMD trials?
Yeah, I mean, I think that's a great question, and I certainly appreciate it. One of the real challenges in understanding whether or not a treatment works in limb-girdle muscular dystrophy is that it's a pretty heterogeneous disease. So there's a wide range of ages and disabilities, and people progress at different rates. And so that means that using old data to see whether or not a new treatment works can be fraught with error. At the end of the day, we don't want to bring a therapy onto the market and give it to a lot of people if we're not sure it works effectively, because these are likely to be pretty big deal therapies and fairly expensive to boot. So many clinical trials require the use of a placebo arm, and that ensures that we're able to really say in a matched group whether or not that drug works. The plus side here is that while it's not guaranteed, many of those same clinical trial programs do offer at the end of the study the active therapy. And so as Dr. Kang mentioned, that would be something that you would really want to understand or hope to understand as best as you could before entering into the study. So some studies have different ratios of the number of people that get placebo versus the active therapy. And some studies make a commitment early on that there's going to be what's called an open label phase where everybody gets the treatment. But no two studies are created the same. And so that's just an important thing that when you're speaking with the investigator in front of you, that you understand, what's the chance you're going to get placebo, and at the end of the day, if there's an opportunity that you might get that drug at the end of the study before it's fully approved.
Thank you. My next question is for Dr. Matthews. Oh, sorry. Did anybody else want to comment on that? Okay. My next question is for Dr. Matthews. I think one thing that everybody wants to know is not just, when will there be clinical trials, but when will there be an approved treatment that anybody can access? I know it's a really hard question to answer, but can you comment a little bit on, are we talking about if there's a clinical trial, might there be a medicine in a year, a decade? What does it look like for other diseases, and what might it look like for limb-girdle?
Great question. And if I had a magic ball, I would rub it right now and give you a good answer, but there's not a perfect answer. We never know because, as has been mentioned before, we don't know whether the things currently in development are going to show effect or no effect. In general, something like about 60%, I think, of studies that get to phase 3 are ultimately approved. If I recall that number correctly. Nick's nodding, so that's about right. So that gives you some idea of probabilities. If you think, and then in terms of the time frame, you think about what has to happen. So let's say a study starts today and needs to enroll 100 patients, and it's a rare disease. And the study, these are often slowly progressive diseases, and let's say we have to go with a clinical outcome measure, and the study has to run for 1.5 to 2 years. So if you say it might take up to a year to get 100 patients enrolled across the country or internationally, and then all of those 100 patients have to be followed for 2 years to get an answer from the study, and then we have to analyze it. So now we're at 3 years, and then we have to go to the FDA and get approval if it's a beneficial, if our analysis shows that it's beneficial. So we are not talking, we're going to have an FDA approved drug at the end of the year, most likely. I extended this, maybe we'll have a good biomarker and we'll be able to answer the question in 6 months, not a year. But the short answer is that in general, we're talking about years, not months, once a study opens. And I'd be delighted to hear other people's thoughts about that.
I think that the other number I like to use, Dr. Matthews, is it takes about, for a rare disease, about 3 to 5 years once you start human clinical trials to get to approval. So,
assuming the drug works.
Assuming the drug works, yes.
Could I just make one other comment about clinical trials in general? I'm going to add one thing to the things that Dr. Kang mentioned that you should ask about, and that is what kind of financial support is there. We've talked a lot about the commitment that families make in terms of participating, and most trials will provide for support for your travel and your hotel and your food, but not your time off work and not your child care. But that's another question that I would say that you should clarify when you're thinking about the commitment of being in a trial. Thank you, Jen.
Great point. Thank you. A lot of the people watching today have subtypes that were not specifically mentioned during the talks today. I was wondering if Dr. Johnson could maybe speak about what's going on with these rare subtypes, or if maybe they would be eligible for treatment with something that's currently being developed for another subtype.
Yeah, I think, I mean, this is a challenge that I know we think about within the GRASP LGMD Consortium. I know that the other stakeholders in the community are considering this as well. One of the real problems with gene replacement therapy is that if you've seen it for one subtype, it's only for one subtype. And there's a lot of subtypes for limb-girdle muscular dystrophy. I think in the future, I'm hopeful that as this path gets paved for some of the more common subtypes, that it will become easier to bring it to bear for some of the more rare subtypes. But each subtype that I didn't talk about has some different challenges. So for the dominant forms of limb-girdle muscular dystrophy, by and large, our goal is going to end up being to knock down that gene expression, which is different than replacing, and something that there's more work that needs to go on in academic labs or other labs that are to think about such a thing. And then for some of the other subtypes, one of the real challenges for gene replacement therapy is to figure out how to fit that size of a gene into that particular virus vector. And I think that's work that, again, needs to happen at the same time. So, and I think there's a lot of hope. I didn't put in my talk, but I know somebody out there would ask a question about it, so I'll say it anyways, about CRISPR therapy, where you're able to more precisely deal with a specific mutation. And there, I think there's still several years worth of work that needs to happen to really understand the safety of that type of approach. So I think, that we should all take heart in the progress that's being made in the subtypes that we talked about today, because those lessons will 100% apply to the subtypes that weren't talked about. But for those other ones, I think we need to work to make those therapies more effective and easier to deliver.
Thank you, Dr. Johnson. Does anybody else want to comment? Yes, Dr. Matthews.
I say one other thing, that I think that some of the therapies are non-specific. So I mentioned myostatin therapy, inhibitor therapy, which didn't help, wasn't shown to be effective. However, there are other non-specific therapies, and some of those may cross limb-girdle types. And it is likely that at the end of the day, we will be combining multiple therapies rather than having one pill that totally fixes things. So even within a subtype, we may be combining things.
I can add one other comment as well. I think as more research is being done on these limb-girdle muscular dystrophy subtypes, we are getting a better idea of what the disease mechanisms are, and a lot of them are connected for different limb-girdle subtypes. So if you recall Dr. Matthews' really nice slide about Fkrp, you could see that there are multiple enzymes related to limb-girdle muscular dystrophy that contribute to that pathway. So I think one thing that would be very helpful in the future would be to find treatments that could potentially apply to multiple subtypes.
Thank you, Dr. Matthews and Dr. Kang. Dr. Mosafar, I didn't get you in there. Would you like a question?
No, I think we've covered pretty much all of the topics, but I think I want to reemphasize what Dr. Kang said, that being in a natural history study doesn't necessarily exclude you from participating in a clinical trial. Now, gene therapy is a different issue because gene therapy may create permanent depot of a drug or a permanent change to the cell machinery. So that is a different issue, but you can always participate in a drug trial, and going from one drug trial to another drug trial requires some period of washout which varies from 30 to 60 days, but you can always participate in that as well. I hope we have that good of a problem where we have multiple drugs and a disease. That would be a good thing to have.
Great. Well, that's all my questions. Thank you, everybody, for the excellent talks and the illuminating discussion.
Thank you.